Yeah, likewise. Maybe before we get going, I can briefly show people how to set up the Google Colab that we're going to be using for this work. So this is actually for the second half of the workshop. We can just quickly take a look. So you can also follow along in the notebook as I do stuff. You can basically just Google for Colab. And then here, right, so basically when you open it right away, Colab gives you a dialog to open stuff. You can go here to GitHub, search for my GitHub handle, which is Grosse-Holz. And you should see this 2024 image processing workshop repo here, among a few other ones. Go away. Ah! Sorry. Google being annoying. Oh, come on. Next try. Okay. So yeah, anyways, open this notebook here. And then sort of, if you run this in Google Colab, so you can also just clone the repo to your local machine. I hope you did that in advance if you want to do that. If you run in Google Colab, you can use this first line here to, yes, we want to run this thing, to clone the repo locally to Google Colab, which will give you all the data and folder structure and stuff, and then install the local version of this library that I copied you here that I checked should work with Google Colab. Otherwise, you basically start from this first cell here and just install dependencies locally. And yeah, I think, should we get going? So you all look very sort of quiet and composed, from which I conclude that many people did this or like have this done already. So anyone who's having any troubles can ask. Also, Alexandra is going to be my TA for this workshop. If you have any sort of technical issues, you can also always raise your hand and she will come answer them. Cool. Okay. So in the absence of people getting completely lost with this, let's get started. Yeah, so as I said, I will run this locally on my machine. Just execute this first code cell to import a bunch of libraries, and then we can start with the intro. So, what are we doing here? This movie that you can see here is a sort of a short snippet of the data that we shared with you. That's an example movie that actually I recorded myself at Institut Curie in the lab of Antoine Comment. And what you can see here is a single molecule dye attached to histones. So what we're doing is we're tracking histones in a UTOS nucleus. And the idea here is to now study the dynamics of genome organization, also genome more broadly, I would guess. So essentially in the first workshop by Alexandra, you learned about PI-C and what it does and how to work with it. And that gives you an overview over the 3D structure of chromatin. And now we want to look at time-dependent effects and how things move, broadly speaking. So for the purpose of this workshop, I will assume that, your awesome collaborator did some experiment and shared some data with you, or maybe you did the experiment yourself and gathered some data. And the data will generally actually look like this. You get a movie that has a few spots moving around. And now the question is, what do we do with this and what do we learn from it? And that's basically the topic of this workshop. There's sort of 3 sections. The first 2 are straightforward, our actual processing of this image. We want to localize these spots, so we want to sort of figure out where they are in each frame of this movie. Then we want to link them together into trajectories. And finally, we want to analyze these trajectories to understand some biology of our system. You can see that that's basically the outline for the rest of this workshop. There's 3 sections. I will presumably focus mostly on the first two, the last one with the analysis. In the current state of the field is a little bit Wild West, so essentially people do whatever they feel like. I put together this third section essentially with sort of analyses I typically do, and to give you a little bit of an intro into what's possible, how one can think about these things. But at the end of the day, a lot of this also depends on what specific questions you're asking. So we will probably skip through this relatively briefly at the end of the workshop. If you have deeper questions or want to know more, always feel free to reach out or talk to me. So let's get going. Localizing particles. So what we want to do is quite literally what's shown in this image here. We want to find the fluorescent spots in our movie. The software we're going to use, I hope everyone has installed, is Fiji. Can I get a quick show of hands of who has worked with Fiji before? Okay, that's many. That's cool. So we can do the intro relatively quickly. I have a few sort of example tasks here for you to play with a little bit and familiarize yourself with Fiji. So the idea is to get everyone a little bit up to speed and on the same level. Given that many of you have some experience with Fiji, we probably won't spend a lot of time on this. Also, I should say, if anyone has any questions on sort of the content here and sort of how to do things, always feel free to ask. I'm very happy to have a live discussion that makes it a bit more live. So let's start Fiji and open up our movie. I put my movie here in a folder that I will also use further through the workshop to generate files and store stuff and whatever. And the first task that we want to do is, as I mentioned, familiarize yourself with Fiji. And yes. So open up the movie. You can zoom in by pressing plus and zoom out by pressing minus. In the first steps, it will increase the size of the image. If you zoom further, it zooms in. So now this movie is still, it looks kind of dark, right? We don't have proper contrast. So you can press Control-Shift-C, or alternatively go to image. Alternatively, you go to Image, Adjust, Brightness and Contrast, and that gives you this nice little window where essentially you can just hit auto to adjust minimum and maximum for the color representation, also grayscale here. And you see this nucleus kind of pop up a little more, and we can clearly see the spots that we're searching. You can scroll through the movie here a little bit and sort of see what's going on. The first thing you notice is photo bleaching. So as the movie goes on, these spots just vanish and the movie generally gets darker. Okay, so why don't we take about 7 minutes to go through these first three problems here. I hope this should be relatively quick. So for the first one, the idea is in this axis down, in this bar down here, you can see my pointer, right? Yes. In this bar down here, you can see your pointer position. So here it tells me x some value, that's in microns. The thing in brackets behind it is the same thing as the position in pixels. And from this, basically figure out what's the pixel size, how big is one pixel in our movie. You might want to zoom in a little bit and get a few positions. You can use the code cell down here to average over a few positions that you look at. Let's take a minute, have people take a look at this. And okay, let's see. Anyone want to tell me what's the pixel size in our movie? Who figured it out? Yep. Yeah, exactly. So the idea is you go to this movie, you zoom in a little bit, you can take a few subsequent values. So here we have x equal up in this top bar, 13.64. I can just plug that in here. And then I can take a few more. So where was it? 6.4, 13.77, 13.90. And so on. And yeah, this spits out 130 nanometers. The true pixel size in this movie is 128.7 nanometers. If you take enough pixel coordinates, this averaging code here will spit that out at some point. As long as you only take a few, it just gives you 130. But that's fine. That's a fair enough estimate for what we want to do right now. So now the second problem here gets a little bit more physicsy. So what are we looking at? We're looking at a nucleus that contains a few fluorescent spots. And our understanding or hope is that each of these fluorescent spots here is really a single molecule. And because this is supposed to be a single molecule, we have a pretty good understanding of how we expect this to show up in a microscope. And that's known as a point spread function. So a single point source of light will show up in a microscope as a sort of blurry blob like this one. And what we expect there is, then the physics expectation is the next task. For here, we basically just want to measure how big are these blobs. So how wide is a point spread function? And for that, the idea is to just find a few points that sort of look nice here, and now we can basically do this by eye. So if I look at sort of this one here, there's a bright one. Generally, these things are sort of about 3 pixels wide. The only thing this code does is multiply by the pixel size we had before, and then print a nicely formatted result. What we get is a diameter of around 400 nanometers, corresponding to a radius of 200 nanometers. And that's sort of this number of 200 nanometers is good to keep in mind. We're going to come back to that later. Now, does that fit what we would expect here? For these single molecule emitters, we have a relationship that tells us how wide this point spread function should be, and that's the one in the next. It doesn't highlight the one. That's the one in the next paragraph here, where lambda is the wavelength of the emission light, the photons that the fluorophore emits, and NA is the numerical aperture that's essentially a parameter of your microscope, which is 1.4 in the setting we had in this movie. So with this, what you usually do is you calculate how wide you expect your point spread function to be. We can now do the inverse here because we just measured how wide our point spread function is. So what we can do here is we can try to figure out the wavelength that this fluorophore emitted at. The idea is you invert this formula, you calculate from the known or measured width of the point spread function, you can calculate the wavelength of the particle. Anyone has a result for me? Or basically take some time, run through this and tell me what you get and whether it makes sense. That's the last question here. That's always a very important question to ask yourself when estimating anything. Does it make sense? Not quite. What did you put for the diameter of the point spread function? But we got up here. We got 0.4, right? So if you take that instead, what you get out here is 546 nanometers. And now the next question is, does this make sense? I think I gave you a little hint here. The fluorophore we have is called JF549. Does that help anyone in figuring out whether our estimate is reasonable or not? You're nodding. That's the statement. Right, seems decent. So the 549 in the name of the fluorophore stands for the emission wavelength in nanometers. So we know that this fluorophore emits at 549 nanometers. By just looking at the movie, we just estimated 546. That's not too bad. I should say this is a little bit lucky because we just put an integer here for the diameter of this point spread function. The point was mostly to get people to play a little bit with Fiji and with these ideas of what are we looking at, how do the physics work. So with that, we can go to the next section here and start trying to find the spots in the state. And what we're going to do here is basically just use this thunderstorm plugin that I gave you, or download it. And the first thing we do is when we zoom out and look at the whole frame of our movie, there's two very bright fluorescent spots down here that are kind of outside the nucleus. That's some sort of dirt. It might be an accumulation of fluorophores in the cytoplasm. It might be some weird autofluorescent stuff in the cell. Either way, we don't really care about this, and we want to exclude it from analysis. So what we do is we draw a region of interest around our nucleus to tell the analysis, look for spots only within this region. To do that, you select the freehand tool up here, and then basically draw, whoops, sorry, I keep pressing random keys. And then draw a shape around your nucleus, including everything that you want to include, and excluding everything that you want to exclude. Sorry. Let's try that again. But yeah, so basically everyone can draw their own ROI. And define your region of interest. So, something like this. So here, for example, you can see there's another sort of faint little spot back here. You might want to exclude that if you draw your ROI precisely. For now, it also doesn't matter too much. So now that we have our region of interest, we can start the Thunderstorm plugin, which is in the plugins menu. You might have to scroll down a little bit and go to Thunderstorm, run analysis. That gives you this main dialog here, which is the control interface for this plugin. The first thing we're going to do is we're going to go to camera setup here and enter correct values for our acquisition setup. The most important thing here is the pixel size, because this is what the software uses to convert between pixels, which is the information it gets, and microns or nanometers, which is the information we want. So getting this correct is pretty important, and also usually pretty easy because you know the pixel size of your movie. The other parameters are used to convert the intensity values in your image. So here, if I go to the image, you can probably cancel this first. In this value bar on the bottom of the Fiji main window, you see there's a value. Each pixel has a value. In this case, it's 3008. And the parameters in the camera acquisition allow Thunderstorm to convert from this pretty arbitrary value to actual photon counts. And that's kind of nice because it allows you to actually estimate sort of localization uncertainty from physics principles and so on. At the end of the day, it turns out that these parameters are not super important for what we're doing here because we're going to estimate the localization uncertainty in a different way. It's nice to get them correct, so you can put these values that I provide. If you have your own movies, you can run calibration experiments to obtain these values. So that's what I'm saying here, that it's a bit, these last three ones are a bit more tricky to come by usually. You can also just set them to default values and just be aware that then you can't trust the uncertainty estimates that Thunderstorm spits out. Either way, this is what we're going to use. And then the rest of these parameters here control how Thunderstorm looks for particles in the movie. So it basically has 3 steps, sort of outlined by these 3 boxes here. First, it smooths the image a little bit to make these individual spots peek out very nicely over the noise, and then it runs some sort of reasonably ad hoc maximum finder on this smooth image. The idea is basically to get candidate estimates for where a particle might be. And once you have those, you then go back to the raw data, so the unsmoothed image, and run a more precise localization estimate for each of these candidates. And that's where here we use an integrated Gaussian PSF. So PSF stands for point spread function. That's also what we looked at before, these individual spots. We basically just assume that it's a Gaussian, fit this thing, and this allows us to get sub-pixel localization accuracy. So you can see all of this is controlled by a bunch of parameters, and we don't really have a good intuition for what any of this does yet. So what we can do is we hit preview down here, which opens up a new window where Thunderstorm shows you in the frame that's currently open in the movie where it would put localizations with these parameters. This measurement window here you can just close. I don't really care about that. Next to that, it also shows you the smooth image. So if you want to play with these parameters for the filtering here, you can see what the smoothing actually does. We're basically just not going to worry about that for now. The one thing I want you to do is to play with with this maximum finding here. So you can see that right now the parameters are set reasonably well. And what you can do is you can play with the prefactor in this threshold here, which controls how prominent a maximum has to be called as a particle. So if I set this to, say, 0.5 and hit preview again, then it starts detecting all sorts of random ground. Yeah, let's take maybe about 2 minutes or something. You can play a little bit with this. You can also, if you want, you can also play with the other parameters and see what they do, of course. Use this preview button down here to check the results of your parameters and find something that looks reasonable. Yes, you can do that in the next step. Okay, so once you've sort of played with these parameters a little bit and checked the effects of what things do, I guess we can move on. The parameters I put here by default that are also reproduced in the notebook, if you're following along there, they give decent results. Yeah, and essentially the way I got these is by playing a little bit with them and seeing what's included, what's excluded, what roughly makes sense. So once we're happy with the parameters that we have, we just hit OK here, and it runs the same thing on all frames of the movie. We can basically just ignore this window here. That's the preview, and now we see that on the movie here, it renders us these localizations. So this looks kind of decent as things pitch out. We still get a bunch of localizations. So here, for example, you can see there's some localization here just in the middle of nowhere. We want to exclude things like that going forward, sort of in the next step. So what we get here is a big table with localization results, and Thunderstorm gives you a fair bit of information. So the X and Y columns are just positions of localizations that it found. The frame indicates the frame of the movie. This sigma here is no, I don't want to solve this. Sigma is the standard deviation of the PSF that it estimates from the fitting. That's quite cool because this allows us to check how big the spot is that it looked at, and we know how big our spots should be, so we can basically filter by this and exclude things that are random noise. It gives you the total intensity of the spot in photons. So this is one of the columns where it would be important to get the calibration of the camera correct for these values to make sense. Because we calibrated it correctly, it's relatively reliable. You can see that per spot we usually get sort of thousands of photons. That's a reasonable value. There's an offset column here, which sort of is an estimate for the background below the PSF that it fitted. You can see sometimes this is 0, which indicates that something went wrong somewhere. And then, like, in most cases, this thing, for example, will also have a giant standard deviation of the PSF. We estimated before, or we sort of measured from our movie that the radius of the PSF should be somewhere around 200 nanometers. This is 600, so this is sort of some big blob that is not one of the fluorophores of interest. Yeah, and finally, this last column here gives you the localization uncertainty that Thunderstorm estimates. So this is a theoretical estimate for how precise it thinks its PSF fitting is and how precise the localization estimate here is. You can see that mostly this is sort of somewhere in the tens of nanometers, which is kind of cool. So now, as I said, we want to filter out things like this, which sort of have a 0 here indicating that something went wrong. They have a giant standard deviation. And that's what we can do down here in this column. So what you can do here is you can, so before that, we use this button here, plot histogram, which allows us to look at histograms for each of these columns. If we look at the standard deviation, we see that we have a bunch of localizations that have somewhere around, so you can see the value on the bottom right, that have somewhere around 100 to 200 nanometer standard deviation. Those are the single fluorophores that we're looking at. And then there's a tail here of things that go up to like almost a micron in standard deviation. So sometimes it just localizes noise. So what we can do there is we can filter for this by saying we only want those trajectories where sigma smaller 500 nanometers. Hit apply. It filters them out in the visualization in the movie, and you can also see down here in this column that it filtered 242 of the localizations. So yeah, why don't you take 5 minutes to play with this filtering a little bit, see sort of what filters you can apply, what you think makes sense. You can combine the expressions in this thing in this line here with logical operations. So you can do AND, offset, greater 0, or something like that. You can also use an OR, you can bracket things, just general logical expressions. So yeah, play with this a little bit, figure out what filtering you think makes sense. You can look into the movie, you can scroll through, see sort of at the end, for example, when photo bleaching kicks in, does it still localize things that are just floating in nothingness? Does it properly localize everything that's there? That's kind of the sort of thing that you want to do. All right, so let's continue. We were looking for filter criteria here, and we do this by looking at the histograms for the different parameters. The first thing I like to look at is the standard deviation of the PSF, because that's usually pretty telling. So here we have a nice peak with most of the localizations being somewhere between 100 and 200, and a relatively, well, fat or whatever, a tail with localizations beyond that. So what I like to do is I like to be relatively restrictive in where I cut this. So in this case, let's cut at like 200 nanometers, for example, which means down in this line here, I say sigma smaller 200. And then another thing that's usually instructive to look at is the uncertainty. So the uncertainty that Thunderstorm reports for how well it can localize things, because spots that it can't really localize, where it knows itself already that it's very uncertain where things are usually either some dirt or just bad localizations, so it also makes sense to exclude those. But we might want to be not as restrictive, so let's cut here at sort of 30 nanometers. Which means we filter like this. And then we can hit apply. And now you can see if I if I undo this filter here and reapply it. So if you take a look at the movie on the right, if I undo the filter, there's a bunch more localizations. If we apply the filter, all of those are filtered out. That's because in the beginning of this movie, there's a lot of stuff going on. Maybe we have multiple fluorophores that are too close together to be resolved, so below the diffraction limit, and that would probably be filtered out by these criteria. If you scroll through the movie towards some later time points, you can see how here sort of the points that it finds always correspond to what we would also by eye see as a fluorophore. There's a few here that it doesn't pick up, so we could still optimize this a bit further. But in the interest of progressing, let's stick with this criterion here. And then one last thing we can do is remove duplicates. So I mentioned to you how Thunderstorm has this two-step approach, right? Where it first identifies candidate particles and then refines those positions in a subsequent step. That can lead to a scenario where you have two candidates that sort of merge to the same localization or the same particle. And of course, we don't want the same thing reported twice, so we can filter duplicates. And what this does is basically, if we put sigma here, it just for any two localizations that are closer together than the standard deviation of the PSF, it will just pick the one. I don't actually know which one it picks, one of them, and discards the other one, so we avoid duplicates. You can see that in this example data, this doesn't actually do much. So it says here it filtered out 5 of 5,000 localizations. So that's not a big deal. And once we have that, we can hit export here. You can put some nice file name here. Let's see. I have to find the folder where I stored everything. Here, so this is my... Right. And now, what I like to do is I just call this... So this is going to save the localizations as a CSV file. So you can look at it with Excel. It's a comma-separated file. You can also load it with Python. That's what we're going to do in the second part. I like to call this just the same as the movie itself, and append something like localizations. So I know that these are the localizations belonging to this movie. So something like this. Exactly, and then make sure here to check this save measurement protocol. What that does is it also next to the, we can actually just check this. So now you see I get two files. One is the CSV file with all the coordinates. We're going to use that for further processing in the second half. And the other one here is something called protocol. That's a text file. And that just contains all the things you did. So here it has the input file name, some rough information about the ROI that you applied. If you want more, you should save the ROI separately. And then it gives you the camera settings that you applied, the different analysis settings, basically everything you did just listed, which is great for reproducibility. Actually, when preparing this workshop, I just looked at how I analyzed these movies and then redid that. Cool. So we now have the localization saved, which means we're at the end of this first part of the workshop, and that means lunchtime. In the second half, we will look into how to link these things into trajectories, and then what to do with those trajectories. And yeah, I'll see you back at 1:30, I think, right? Cool. There we go. Cool. All right. Welcome back. Hope you guys had a nice lunch break and are back here full of energy and ready to learn how to link together trajectories and then deal with them. So that's the two sections left in this workshop. I'm going to walk you through them over the next hour. And yeah, let's see what we can learn from this movie that we looked at in the first section. So for the remainder, for these remaining two sections here, I will be working in this Jupyter notebook here. So I hope you can open it on Google Colab, as I showed you in the beginning, or on your local machine, whatever works best for you. We can basically just close everything associated with the movie itself and get going. Oops. with our notebook. There we go. So let's build things into trajectories. For anyone who didn't sort of finish the first section where we saved the localizations, I'm providing a backup file. So if you look at the Git repo where we shared the original data with you, there is a folder called backup intermediate files, and there is this localizations.csv file that you can use if you didn't generate it in the first half of the workshop. So yeah, let's basically start by sort of setting up environment. In this code cell here, it's basically just setting up some variables that we're going to use throughout the rest. It's not that important. The thing to get right is this thing here. So we need to know where the file is that we want to load the localizations from. And we also need a sort of directory where we can sort of store output and all those things. So the way I set this up on my machine is I have this folder called processing here. So here there's this backup folder, for example. I have this folder called processing. There's the movie in there. That's also where I stored my, here's the CSV. That's also where I stored the localizations. And that's what I'm putting in this working dir variable here. And then I just tell the code that the localizations to load are this file in that. Yeah, as I said, the rest is not particularly important right now, so we can just run this cell. And the one below it will actually load the data and sort of show the head of the data frame. So now we loaded the data. And we can see that this is basically exactly what we saw in this table in Thunderstorm as well. The columns are basically the exact same thing. We just now have it loaded as a pandas data frame in our Jupyter. Anyone who does not sort of see this thing pop up can sort of raise their hands, and Alexandra, for example, might come by and come talk to you people. Okay, I see a few people, so I'll also go walk around and troubleshoot a little bit. Let's take sort of 2 minutes to get this going. All right, so it sounds like most people figured out how to get the data into Colab or the local machine and load it. So let's continue. First thing you should always do if you look at a data set that someone shared with you or you generated yourself or whatever, is just run a few sanity checks. Make sure that what you assume is true about the data set is actually true. We will do two things here. The first thing is we just look at how many localizations do we get in what frame. So in this plot here, on the x-axis you see the frame of the movie, so it's time basically. On the y-axis you see the number of localizations we get in this frame. You can see that the number of localizations decreases sharply as the movie progresses. There is this one spot here that just shows up until frame 400 something. And this matches with what we saw visually in the movie, so that's good. That means that our localizations are probably reasonable. Next thing is already a bit more subtle. This is subpixel bias. So I told you that, if you remember, I don't expect you to remember these numbers. That's why I will give them to you again. The pixel size in our movie is 128 nanometers. Thunderstorm reports that it can localize our fluorescent spots with an accuracy of 10 nanometers. That's an order of magnitude difference. So we better make sure that in reporting these very accurate localizations, Thunderstorm didn't actually get confused by the pixel grid that's overlaid, but interpolated everything correctly. One issue that could happen is if this subpixel localization, for example, screws up for some reason, it might just put all the localizations, say, in the center of a pixel, or sort of on the edge, or something like that. So one quick check we can run to see that at least there's no obvious things going wrong is to just take the sort of part after the decimal point of the localizations in pixels and plot a histogram. And that's exactly what this code does here. And the histogram you get here, you can think of this as basically one pixel with all the post-decimal point things of all the localizations histogrammed over it, and we see that this is basically uniform. There's a bunch of noise, that's fine, but there's no sort of discernible pattern in here, which means that we can at least reasonably trust this subpixel localization. There's nothing obvious going on. Once we've convinced ourselves that our data set is not obviously wrong, let's get into actual linking. So the problem we're facing is that Thunderstorm doesn't give us trajectories, right? It doesn't tell us particle 1 is at this point in one frame and at this point at in the next frame. It just gives us localization in individual frames that have nothing to do with each other. And now we have to do this linking together, sort of identifying which localization in which frame belongs to the same particle. We're going to do this using the TrackPy library. So at the end of the day, this is going to be essentially one library call. But of course, it's important to understand what's happening behind the scenes. So the figure I put up here basically gives you a little bit of an intuition. You start in some frame of your movie. You have a bunch of localizations. That's these little stars here. And essentially what you do is you draw a circle around it and say everything that's in the next frame within this circle around one of my localizations is a candidate for the same particle in the next frame. So that's kind of the figure over here. And if your imaging conditions are very nice and easy, then you will basically find one particle within this linking distance, and then you can just link them together and be happy. Of course, that's not always going to be the case. Sometimes you find multiple particles in this linking cutoff, and then you have to make some decision which one to choose. And essentially, like there are a bunch of different algorithms to do this. The simplest thing you can do is just take the closest one. If you have a lot of these events in your data, that's going to introduce some bias, so you have to be careful about it. In the example data we have here, labeling is sparse enough and the loci move slowly enough that we basically don't really have this issue, which is quite convenient. The other issue that could happen, so now we went through what happens if you have exactly one localization in this cutoff radius, what happens if you have more than one. Next thing is what happens if you don't have any. Then basically you lost track of your particle, so the trajectory ends, and you just cut it off there. Because because we were kind of conservative in choosing our filters in the localization part, it's possible that the particle is actually still there and is still emitting light. It just, got blurred for some random reason, and we kicked it out in the localization step. So maybe we just kicked out a localization that should be there. An alternative is that some fluorophores blink. So sometimes they just transition to a dark state for a few frames in the movie, but then they show back up again. And you basically don't want to chop your trajectories just because your fluorophore disappeared for 2 frames or something, because that would give you many very short trajectories and only very few sort of really long contiguous trajectories. So the second thing to pay attention to when linking these trajectories is something called the memory of the linker. So basically, how many frames a particle is allowed to disappear for before you actually say, okay, this is where the trajectory is. And what we're going to do now is basically try and identify reasonable settings for both of these parameters. So the 2 parameters are the linking range, how far do we look for particles in the next frame, and the memory, how long do we allow things to disappear. The thing we're going to start out with is the search range. So how far do we allow a particle to move? And this is a bit of a tricky situation because how far we allow a particle to move between frames depends on how fast it's moving. If you're tracking very fast particles, you can expect that between 2 frames, they move like 100 nanometers or something. If you're tracking very slow particles, you probably expect that they move less. So yeah, to some extent, there's a chicken and egg problem where, of course, the trajectories you get out at the end and the results you can, sort of the insights you gain from those trajectories depend on this linking setting. But then which setting to choose also depends on what your particles actually do. So how do we figure this out? One thing I like to do is to basically look over one frame, how far do my particles move? And of course, I can't do this I can't really do this for now because I don't know which particles in the 2 frames belong to each other. But what I can do is just calculate all the distances that we have and always take the shortest. And that's basically what this code here does. So if we execute the code cell, we get the distribution for each particle in each frame, the closest distance to any localization in the next frame. And what you can see here is that this is clearly very much bimodal, because what's happening is that if the same particle exists in the next frame, there will be two localizations across these two frames that are very close together. So if you look at the closest distance for the localization in the first frame, in the second frame, it will pick up on the one very close by. That's this first hump. The second hump is the situations where we don't have the same particle in the next frame, and then the closest distance from the localization in the first frame goes to some random particle, reasonably close by, but not as close as it would be if it was actually the same particle. And what you can see here is that these two modes are very nicely separated in this example. In other data sets, this might be a bit more smeared out. Here, this is very clear because the labeling in the image is reasonably, and the movie is reasonably sparse. So this is a kind of nicely principled way to set a linking distance. So here we can, if we look at the x-axis, we can set like 300 or maybe even be a bit more conservative and set it at 200. It's not going to make a lot of difference. Note that the y-axis in this plot here is also log scale. So basically, this thing down here is like 2 units. Okay, so that gives us a good estimate for what to do with the linking distance. And now the next question is what to do with the memory. And I'm basically not going to tell you how to figure this out. What I want you to do is this exercise 1 here. Basically run the linker with settings you can give here. So you see what's happening here. This is, as promised, this is the one library call, trackby.link. We give it the data frame with the localizations that we loaded from file. We tell it where to find the positions for the particle and which column in the data frame indicates time. And then it has these two parameters that I've been talking about. And what you can do is you just run this, and it spits you out a data frame that looks like this, where now you have an added column here that identifies particles. So here, because this is the head of the data frame, so we're always in frame 1, all the localizations are different particles. The localizations in subsequent frames will now also have some particle ID associated, and all the localizations with the same ID belong to the same frame. So yes, exactly. Exercise 1. Use this code, play a little bit with this memory setting, and think about how would you identify a reasonable setting for this variable. Let's take about 3 minutes or something to play with that a little bit, think a little bit, and then. All right, so there were a few questions about the path if you work in Google Colab. The path where sort of the base of this directory structure you see in the left column, that's just a dot. And then you can sort of / put folders where your data are, or just put stuff in that base directory right away. Okay, so I hope people were able to run this linker. Does anyone have results they want to share, or like thoughts they have on how to determine this memory variable? What should we do with that? All right, doesn't sound like we have volunteers. Then let me walk you through what I would do. What I generally do is basically I just run this with a few different settings, and then look at the results. So here I'm saying, okay, let's do search ranges of 200 or 300 nanometers, and run the linker with a memory of 0, 1, 2, or 3 frames. So this is going to be a total of 8 runs of the linker, and then we're going to look at the results. So this takes a few seconds to execute. And then what I'm plotting down here are the survival curves of the trajectories. So what you see here, on the x-axis is the number of localizations in any given trajectory. So now we're working with this data frame we got back from the linker call. So now we have identified which localization from Thunderstorm belongs to which particle. And what I'm plotting here on the x-axis is the number of localizations per trajectory. So how many frames do we have? And on the y-axis is a cumulative count. So basically, how many trajectories are longer than the value indicated on the x-axis. So this thing will always monotonically decay by construction. And it's essentially, for those of you who are familiar with it, it's just 1 minus the cumulative distribution function. And basically how fast it decays tells you how long your trajectories are. First thing we see here is that if you look here, for example, there's a blue and a purple curve right on top of each other. If we look into the legend, blue is search range 200 nanometers without any memory. Purple is search range 300 nanometers without any memory. And if you go through the other curves here, you will always see pairs of curves that basically lie on top of each other. Which means that the search range setting, it doesn't really make a difference whether we choose 200 or 300 nanometers, and that's exactly what we expected from this bimodal distribution that we saw. There's only very, very few sort of steps that fall in between this gap, so whether we cut that off or not doesn't make a difference. The thing that does make a difference is the memory in the linker. We see, so here I pointed out, these are the curves without any memory, where we only accept really contiguous trajectories, and if we accept a gap of 1 frame, this curve jumps up a little bit, and we get a few longer trajectories, which is nice. We generally kind of want our trajectories to be as long as possible. And you can see if we allow the memory to be even longer than this, it actually kind of converges. So it also at some point stops mattering too much. There's still a little sort of jump here. So what I do in this example is I set the memory to 2 frames. That kind of seems to be the point where things converge. So just setting it to 2 frames is basically the most conservative thing we can do while still taking everything that we're reasonably sure of. Cool. So that's basically what this paragraph here says. So let's do that. In this next code block, I have this variable use linking where I basically just identify which of these settings I want to use. You can essentially choose whichever one you're happy with. This is the one I will use. And then what we also do is we restrict trajectories to have a minimum of 3 localizations. Everything shorter than that is basically just random noise popping up somewhere. And you can actually see from these survival curves here, we do have a fair number of those. So the first data point here are trajectories with a single localization, which means it's one localization that never came close enough to anything else to be linked into a trajectory. We're just going to kick those out. That's noise. The rest of what this code block does is it now changes the structure of the data that we're working with. You saw when you ran the linker, what the linker itself outputs is a data frame that just assigns an ID to each localization of which particle it belongs to. So that's cool, that contains all the information that we need, but it's not really how we kind of think about a data set of trajectories, right? So that's basically what's happening in this block here. We're reformatting the data in a sort of somewhat more intuitive way, where then we really have a pile of individual objects that are trajectories that we can deal with, that we can query for like how many valid frames do you have, how long are you, all these things. So this is an object-oriented data structure that comes from this library Noctiluca up here that you installed in the first code. Let's do this. It outputs a little message where it says that we have about 300 trajectories that we got from this movie, and which linker settings we used. So here there's a bit of a wall of text on how these objects that we just created work. Because also, full disclaimer, this is a library that I wrote to deal with trajectories and fluorescence microscopy datasets. So I don't expect you to be familiar with it. But I like using it because it has this sort of intuitive idea of, we have a pile of trajectories that then can kind of pull out the things we want to work with. So to get you a little bit familiar with how these things work, I have 2 little exercises here. They have given solutions, so you can also just execute that if you want to. But if you want to get a little bit familiar with what's happening here, there's basically a few basic questions to look into what our data looks like. So what's the average fraction of valid frames per trajectory? our linker now allowed a maximum of 2 frames missing in allowed gaps in trajectories of a maximum length of 2 frames. But if our data consists of trajectories that have 1 localization, gap of 2 frames, 1 localization, gap of 2 frames, 1 localization, gap of 2 frames, that would be kind of unfortunate. What we're really hoping for is that these gaps are not particularly common. That's basically the idea of this first question here, to just check how many frames in the trajectories are actually missing. Are short trajectories sort of better or worse on this? You can plot the survival curve and compare if you plot the survival curve as a number of valid frames in the trajectory, so localizations that are actually there, or as a number as a function of length in the trajectory, so sort of how much time essentially the trajectory spans. And the final one here is to plot a histogram of the uncertainty that thunderstorm reports. So let's take about 10 minutes to do this. Let's see, actually, yeah. Let's take about 10 minutes to go through this exercise 2 here. And exercise 3, I will also show you afterwards. If you want, you can also do this yourself. But then we're going to move on in 10 minutes. Exercise 3 is the more interesting one for moving forward, but exercise 2 is kind of the basics of how to use any of this in practice. All right, so in the interest of time, I think let's move on. I see people are still working concentratedly, I guess. But yeah, let's move on. So let's quickly walk through how these things work. First, we want to calculate the fraction of valid frames in the trajectory. As indicated in this bullet point list here, you can query a trajectory for the number of valid fractions. Number of valid frames or total number of frames with these calls. So this is so you basically just divide one by the other, and what we get is that about 80% of our of the trajectories are 80% of the time points on the trajectories are valid localizations. That means about 20% are missing frames. That's kind of we can stratify by long and short trajectories. We just set an arbitrary cutoff of 10 frames. And we see that the fraction of missing frames in short and long trajectories is very similar, which is a good sanity check, because that means that the missing frames are, at least, with this very rough analysis, it's consistent with the missing frames being independent of trajectory length, which is good. We would be worried if it was otherwise. We can again plot survival curves in terms of total number of frames or number of valid frames. You see these track each other very closely. That's also nice. And finally, we can plot a histogram for the localization uncertainty that thunderstorm reports. So this is basically just some meta information on each trajectory. We can histogram this. That gives the blue histogram here. In orange is the histogram of means over trajectories. So the fact that they have similar spread here hints at the fact that the localization accuracy might actually be heterogeneous across trajectories. We would have to run some deeper analysis to get a really reliable statement on that. So now this exercise 3 here is also kind of fun. The idea is to basically just take a look at how our linking results work. So we're just plotting all the trajectories, and here what I'm plotting is always each single coordinate in the trajectory versus time. And what you can see is that overall this looks pretty decent. We have like this trajectory here, for example, which is sort of just nicely detected and linked as one trajectory. There's also things like this thing down here where we have multiple shorter trajectories that are sort of very much right behind each other. And it kind of looks like this is the same particle. We could go back into the movie to check whether it actually looks reasonable to link these together. For now, we're going to be happy with saying we see this here in the coordinate 1. We also see it in coordinate 0. So very likely these things are just actually the particle disappearing for something. As an example of a post-processing step, what I put in here is basically a step to relink across these larger gaps. We can basically just execute that for the time being. If you're interested, I can walk individual people through what's happening here in a bit more detail. The deal is that after we do this relinking step, we now only have 180 trajectories. So apparently there were a few relatively short ones that now got linked together into longer ones. It's kind of nice. And then at the end of the day, again, we filter for trajectory length, where we kick out everything that's shorter than 10 frames. Those are kind of not really useful in the kind of analysis that we want to do later on. So at the end of the day, we're left with about 100 trajectories that have decent length. And that's a pretty cool data set, given that this all comes from a single nucleus that we observe in the microscope. So, of course, in a real scientific setting, if you want, you would record more movies and you get a lot more data than this. But for now, we got 100 trajectories out of this one nucleus. That's pretty cool. Before we conclude the second session, the second section, and transition to the analysis, let's do one analysis step already and just plot an MSD curve. So this code does that. You can look into it if you want. At the end of the day, this is the plot that we get, where each colored line in the background is a single trajectory MSD, and the black thing is mean square displacement for the ensemble of trajectories that we linked from our movie. So this is one of the things we want to look at in the analysis section in a bit more detail. So let's get into that. What we do to get there is we save our trajectories as a file. We add a little description saying sort of what's in this file, how do things work, and that's the section on linking. So now we have about 15 minutes left to quickly look at how one might analyze these trajectories. As I mentioned at the beginning, this depends a lot on the biological questions you have and sort of what you're interested in. So that's why I'm sort of going to breeze through this a little bit and show you sort of some of the standard things I would do. This also, again, depends a little bit on code that I wrote in my PhD. If you have any questions about it, I'm very happy to chat. This is some of the things I do. Sort of this section is more closely related to what I do in my own research. So similar to the linking section, in the linking section we wrote our trajectories to file, which means we now can just load this file again. And again, if you didn't manage to go through the linking section, there is a backup file provided in this backup intermediate files folder. Take the one with the .h5 ending. This is HDF5. It's a nice file format. So similar to the linking section section, whenever you load a data set, first make sure that it kind of looks reasonable and has the features that you expect, also at least doesn't look awfully wrong. We're going to plot some sanity checks. First is the survival curve that we already looked at. We see that we have a bunch of trajectories that are, everything's longer than 10 frames because we conditioned on that. We have a few trajectories that are longer than 100 frames, which is kind of nice. Overall, this looks decent. We can plot this MSD plot again. Still see, it's the same plot as before. It looks relatively homogeneous. It's not like we have sort of clearly two clusters of trajectories here. It looks relatively decent. That's nice. So, having plotted this plot for the second time and not even told you what it is, here is an explanation. Again, it's a bit of a wall of text. You can go through it on your own if you're interested in the details. The gist is MSD stands for mean square displacement. So what we do is we take the trajectory value at some time, and at some later time point with a defined lag time, which is delta t in this equation here. We calculate the difference, we square it, and we take an expected value over the whole trajectory, and in this case also over the ensemble. Sometimes in the literature they call this time average MSD because you're averaging over the trajectory. I will just refer to it as MSD because this is basically what the only thing we're looking at. Now, what this tells you is over a time window of a given length, how far on average does your trajectory move? So I plotted a few examples down here. What we call a ballistic trajectory is something just moving in a straight line. So this thing here, because it's moving in a straight line, would have an MSD that is proportional to the time lag square, because the distance you move is proportional to time, it just goes in a straight line, which means if you square it, which is what we do in this equation here, you get a dependence on, you get a scaling with time squared. In standard diffusion, so if you take a random walk, a particle that at each time point just takes a step in a random direction, that's diffusive motion, and characteristically has an MSD that scales linearly in time. I have some explanation up here for why that is. For now, the other interesting example is white noise. So if we fix a point in space and then have a sort of quote particle that just in each frame we take in our movie appears in a random location around this point, that's going to look something like this, and that has a flat MSD. You can calculate what it looks like. If the standard deviation of this thing here is sigma, then the MSD will have a constant value of 2 sigma squared for all time lags greater than 0, and it's always 0 at 0. The last interesting thing is what do more complex objects like polymers, for example, do? If we track a single locus on a polymer, like for example, a histone on DNA, we expect this thing to kind of want to diffuse, so it gets kicked around by thermal noise, but at the same time it's tethered into this longer chain of polymer. So if it tries to move somewhere in a given time point, in the next time point it will have to move back. So it's not completely confined as this white noise example here that I have, where it's really sort of confined in a single volume, but it's kind of more constrained than free diffusion, right? So what we expect is that the MSD of a locus on a polymer is somewhere between the one for free diffusion and a white noise. Trajectories for such a process look something like this. It basically looks like what I just told you. It's a bit of a mix between this white noise thing and the diffusive one. It's more compact than the diffusive, but at the same time not actually localized in space like the white noise. And the MSD we generally expect to scale as the time lag with some exponent. This exponent 0.5 that I put here is what we would expect from the Rouse model, which is a specific polymer model. But yeah, generally, sort of these are sort of the types of motion you can expect to see in single particle tracking. Oops, sorry. And yeah, so the first question here is, if we look at our MSD plot above, what type of motion are we looking at? Here's the plot. Anyone want to venture a guess? So here, as I tried to explain in this in the section down here, we're basically trying to look at the exponent in how the MSD scales, which in this plot here, because it's in log-log scale, is just the slope of this curve. If you sort of eyeball this a little bit, this spans about 2 orders of magnitude in time, and that gives us about 1 order of magnitude in distance. So that's a slope of 0.5, which means this is this exponent in the MSD would be around 0.5. That's basically what we expect for polymer motion. So that makes a lot of sense. We're looking at a polymer. That's great. Now, this, of course, is, you can eyeball your data, but you can also try to be a bit more precise. There's two things that affect the MSD quite significantly, and sort of two error terms here. One is localization error and one is motion blur. Localization error means that your localization algorithm, in our case Thunderstorm, will of course not be able to exactly with angstrom precision pick up the localization of your fluorophore, much less the location of the actual histone, because that's not where the fluorophore is. Anyways, the point is your localization will always be a little bit off, and this basically adds white noise to your MSD. Yeah, we'll go through the logic down here. The second effect is motion blur. The movie I gave you is recorded with a lag time between frames of 125 milliseconds. And to capture enough light from the fluorophores during this short time, we actually had continuous exposure, meaning the image you see is integrated over these 125 milliseconds. It's not that you sort of take a snapshot and then you wait for some time. You just integrate everything that comes in over this time, then you write it out and keep recording more. Which means that everything that moves during these 125 milliseconds will be recorded as a kind of blur in your image. You usually can't really see this by eye, but it does affect the MSD if you calculate it, as we will work out in the questions here that I will basically just walk you through. The neat thing about motion blur is that it sounds kind of annoying to deal with because it sort of mixes things together across timescales where you don't really have experimental data. The point is we can just calculate what this thing does to our MSD. This is the expression, it subtracts a constant. And this is the constant if your MSD is a power law with some exponent output. So what you can do is basically take these two equations and rearrange them such that you get an expression for the real MSD in terms of the motion blurred one, which is of course the one that we observe. This looks something like this. So there's a bit of math. At the end of the day, we can calculate what we expect this constant to be, and then we can just add it back to the MSD that we observe. We will do that in the code below here. Before that, let's briefly talk about localization error. As I mentioned, localization error means you basically add white noise to your MSD. I mentioned before that white noise has a constant MSD, and because these two processes are independent, the error you make in localizing your particle is hopefully independent of the actual motion of your particle. That means you can just add the MSDs, basically because MSDs work like covariances. Covariances of independent variables add. So that's why to add localization error, you just add this offset with the localization error. So let's see how this works. Here is the ensemble MSD. So the thing that was in black on the plot above is now blue. That's the raw data, sort of the MSD of the raw data. And then in orange, we see the same curve now with this constant for motion blur added back in. And you see that it actually significantly changes things. We then subtract localization error, where here we basically just use the value that Thunderstorm gives us. We plotted the histogram above. Up here, we saw that Thunderstorm reports localization uncertainties somewhere around like 12, 15 nanometers. And I basically just used that in this analysis here to subtract localization error, essentially to make the point that localization error is a smaller contribution than motion blur in this specific data set. Of course, in any other data set you have, that might be different, and either one of these contributions can be the more important one, or maybe both of them. So now, having seen that taking these errors into account correctly is important, I also basically just showed you how to do this by hand, which is a bit approximate. If you look into the text that I wrote here, to calculate this motion blur, you actually have to assume that the exponent is 0.5, which of course we actually want to fit. So we basically have to do all of this at the same time, which is where we again resort to library code. So one more point in analyzing MSDs is that the error you get on this curve is highly correlated. What that means is that, so here at the end, you see how this curve kind of becomes a bit jaggedy, and that just looks like noise. We probably wouldn't really trust that. But up here, this hump that it does, that still kind of just visually looks like, the curve is still kind of smooth. It kind of looks like an actual feature in the data. And let's so the issue is that this MSD curve will sort of look visually relatively smooth, even in regimes where you're mostly looking at noise. To check that, we can bootstrap this and basically just look at ensemble MSD curves for randomly chosen halves of our data. So what I'm doing in this code block here is I'm randomly selecting half of my data set and then plotting an ensemble instead. And you can see how these curves diverge way earlier than what you would expect to be noise from just looking at sort of visually, how smooth does it look or something like that. That's exactly this effect of highly correlated errors. And it also means that if you just take this plot and you fit a line to it, you're going to make a significant error. Or like your estimates will be biased. So to avoid that, you can do your statistics a bit more rigorously. And yeah, this is so here it basically says, let's run the code below and figure out after what's happening. This is again a library that I wrote during my PhD. It fits these MSDs essentially, gives you a bunch of parameter values, and correctly takes these correlations into account. And the last thing is this one, which gives us error estimates for this fit, which is always kind of important. So here we got we get an estimate for this alpha. So basically what we're doing is we're fitting a power law to our MSD, taking into account motion blur, localization error. You see that it's actually fitting localization error. And at the end of the day, one of the things we're mostly interested in is this exponent. We get a value of 0.478 something, which by itself is kind of meaningless because we don't really know what's the error bar on this value. So, for example, I told you above that a Rouse model would have 0.5. Is this significantly different from 0.5 or not? That's where we're calculating the error bars down here. This is going to take some time to run. Should be done in like 30 seconds or something. In the meantime, I will point out that, so what just happened? What did we do? There's a bit of theory behind this. At the end of the day, what we're doing is we're fitting a Gaussian process to our data. If you want to know more about this, hit me up. Always very happy to walk people through this. So in the meantime, this run finished here. We get credible intervals for our parameter estimates. And for the exponent, we see that the credible interval is 0.42 to 0.54. So our sort of expectation of 0.5 is in this interval, which means the data we have is perfectly consistent with the Rouse model, which, by the way, doesn't necessarily mean anything biological because the Rouse model is not the most physical model you can come up with. But it's kind of a fun observation that it's consistent with the data that we have. Okay, one very brief last thing I want to do is, if you go through this math of fitting Gaussian processes to your data, you end up with basically one process that can generate data for you. So you can not only fit it, you can also use it as a generative model, which means we can do a fun thing where we check trajectory level heterogeneity. So here's our trajectory-wise MSD plot again, where we see that if we look at the single trajectory MSDs, these colored lines here are scattering about an order of magnitude, which we would expect some scatter there because we have finite trajectories, we have finite data, there's going to be noise, it's going to scatter a little bit. But will it scatter this much, or is this more than what we should expect? Because we have a generative model, we can check that. We can basically just use the process we fitted to our data to generate a data set that matches our real data in terms of trajectory length, number of valid frames, and so on. And that looks like this, where we see that the scatter we expect in these single trajectory MSDs is a fair bit less than what we see in the real data. So this allows you to make a statement about heterogeneity, where you say that some of the loci we're tracking are moving faster than others, which in a biological sample is maybe not particularly surprising, but that's where it gets back to a research question: why do some histones move faster than others? And that's where I think I'll have to leave you guys. Thank you very much for listening. Of course, yeah. As I said, happy to answer any questions. I think we have a coffee break, right? Half hour or 15 minutes? All right, so 15 minutes coffee break, and then we'll get a workshop on machine learning.